Hello viewers, welcome once again to our ongoing series on sex determination mechanisms, which is falling under the broader genetical series of Mendelism and beyond. In my last lecture, I had discussed with you the implications of the XY-XX system and the ZW, the ZZ and the WZ systems of classification of the sex determination. We had also discussed about the XO systems which was prevalent in insects. On your screens, viewers, you would find a summary of these three type of the sex determining systems. Now we ask ourselves a question: are these the only types of mechanisms that determine sex? The answer is no. There are many more. Let us see some of them in today's interaction. There is a case of multiple sex chromosomes, because we were till now talking about only one X or two X or X and Y, but then we find that in some dipteran insects and many dioecious plants, we have more than one pair of sex chromosomes. And a classical example is seen in a duck-billed platypus. You know, Ornithorhynchus is a blend of mammalian, avian, that is bird, and reptilian characters. In fact, it is an egg-laying mammal. And 2n, the diploid number is 52, and there are 5 pairs of sex chromosomes. Interestingly, the females have 10 X chromosomes, the males have 5 X chromosomes and 5 Y chromosomes. So the complete genome of this platypus has been sequenced, and it has been found to have 2.3 billion base pairs of DNA, and it comprises 18,500 protein encoding genes. Now this egg-laying mammal would have the multiple sex chromosomes, which you can see on your screens. They are there in a sequence which make the homologs. So it is not one X or one Y which is determining sex in this case, it is multiple ones. We also have a mention of compound chromosome systems in literature. So that means instead of just one X or a Y, there could be a compound chromosomal situation, because there are 8 X chromosomes, 12 X chromosomes, and even 6 Y chromosomes which are present. So in this case, how can you expect that the chromosome number in males and females would remain constant? It would not, and there will be large differences in their number. The example of a nematode Ascaris is a very familiar one, because there are 8 X chromosomes and 1 Y chromosome in the genome, and in addition the species has 26 autosomes. So that means in all it makes 26 plus 8 plus 1 Y, that is 35, and this is the situation in males. But in females it is 26 autosomes, all right, but the X chromosomes can vary from 4 to 16. So that means the chromosome number can go up to 42. So there is a lot of difference as far as the chromosome number between the males and the females is concerned. And in case of a beetle, that is the Blaps beetle, we find that both the Y chromosomes and the X chromosomes are present in compound forms. In Blaps, we found that the male has 12 X chromosomes, and the Y chromosome number is 6 or even more, and this is in addition to the 18 autosomes. So there are whole lot of situations that may occur as far as the compound chromosomes is concerned. But then next, when we see a situation of sex determination, it is very, very interesting, because in some insects there are no sex chromosomes at all, and yet there is a perfect mechanism of sex determination. In this case, we find that the sex in these insects is not based on sex chromosomes, but it is based on the number of chromosome sets which are present. That means the question here is whether an organism is haploid or diploid. So it is very rightly called as male haploidy or haplodiploidy. So that means unfertilized eggs, they are there, they will develop into haploid individuals, and they are going to be in almost all the cases males. Now this is a very different type of a parthenogenesis, and diploid individuals are generally female. They could even be diploid, and yet they could be sterile males. So we find that the cells of the males are possessing only a single set of chromosomes which are inherited from the mother. So that means they are haploid. And in contrast, the cells of the females will possess two sets of chromosomes because they are diploid, and one set naturally would be inherited from the mother and the other set from the father. On your screens you would find an example of a haplodiploidy system, where you find that the diploid is 32, which is a female, and when the haploid number is 16, then it is a male. In other words, the males are always going to develop from unfertilized eggs, so they will be haploid. And a cross between the parental generation of a female, the female would have two types of eggs. If they are fertilized, fine, they would give rise to a female. If they are not, that means they would give rise to a male, because the zygote is not there, and if at all it is there, then it has to be a male. If you see on the right hand side of a male, that is a haploid, the gametes of this male have to be mitogametes, because meiosis will not take place in haploid organisms, and therefore as a result of mitosis the sperm would be formed. So the half of your genes in case of a diploid normal genetics, we would find that there is 50% chance of receiving the same allele from each of the parents. However, in case of these insects that are showing haplodiploidy, the males are going to be producing mitogametes, and therefore the same set of parental genes will be got from the paternal parent. Whereas the diploid females in these insects, they are absolutely normal because they are showing meiosis and crossing over. So if we look at the sisters of these, there are 50% chances of receiving the same allele from their mother, and obviously 100% chance of receiving the same allele from their father, because it is a case of mitogamete, that is, the sperm is formed there. So the average relatedness between the sisters is 75%. As you can see on your screens, the relationship of the different types of chromosomes, or the kind of chromosomes which are coming in both the daughters from the mother, have been depicted. Whereas if we talk of the brothers have a 50% chance of receiving the same copy, each from the mother's two alleles at a particular locus. So their relatedness is only 50%. Why are we talking about the relative relatedness of the brothers and sisters? Because the greater genetic relatedness among female siblings in insects, which are determined by the haplodiploid sex determination, it really contributes to the high degree of social cooperation that exists among females, that is the worker honeybees or worker ants of these insects. On your screens, viewers, you will find another example of a wasp. This wasp is a Brecon habitor, which again shows different chromosomes. In a diploid we have 20 and in the male we have 10. So again it's an example of a haplo diploidy system. And in most of the cases in ants and males, we find that there is no sex chromosome, as I said before, and rarely diploid males can also be formed. But then they will be very low in viability, they will not give rise to viable offspring, and most of them are even sterile. And even if they can produce a viable sperm, this sperm would be then diploid, and because it will be a mitogamete, and then it would further give rise to a polyploid series, that is a triploid series, and again the progeny would be sterile. Let us take a classical example of honeybees, which fit perfectly well into our example, as I said, of 75% relatedness of the sisters. Now, in this case, it's very fascinating because the different types of bees are assigned with different jobs. There is a queen bee, worker bees, drones, and others. So what is going to control this mechanism? Perhaps it is the nutrition also. It is here that the royal jelly becomes the key factor. On your screens you would find that the larvae of the developing queen, they are surrounded by a special type of a substance which is called as the royal jelly. Actually, this royal jelly is secreted from the glands in the heads of the worker bees, and it is fed to all the bee larvae in the beginning, whether they are destined to become drones, that is the males, or whether they will become the workers or the sterile females or the queens, which are the future fertile females. But then what happens is this nutrition is stopped after 3 days. The drones and the worker larvae are starved. They are no longer fed with the royal jelly, and a discrimination is made. Only the queen larvae continue to be fed on this special substance throughout their development, which leads to the formation of a different type of morphology, including the fully developed ovaries, which are obviously needed to lay eggs. And it has been found that these royal jelly components include what is called as major royal jelly proteins, abbreviated as MRJPs. So they are the kind of proteins which are synthesized by the bees. And there are about 9 types of different proteins, and in addition this jelly has simple sugars, fatty acids, 10-hydroxy-2-decanoic acid, and many water soluble vitamins, antibiotic elements, and other trace elements. If we look at the screens, viewers, you will find that the queen honey bee would have, as a result of meiosis, the haploid number, gametic number is 16. So there are 2 possibilities. One, it undergoes parthenogenesis because it didn't have a chance to mate or to get fertilized, and this parthenogenesis gives rise to a male, and the number of the chromosome number of the male would remain 16. And if by chance this particular gamete, that is the other haploid gamete, it fuses with the male or the drone sperm, we would find that now fertilization would result in the formation of a female which has a diploid 2N. Now again it would depend whether this female has been fed on royal jelly or not. On the extreme right you find that it is fed on royal jelly, it becomes a queen and again it is fertile. And if there is no royal jelly feeding, then it remains a worker and it is sterile and it will do all the other household chores. So in this diagram you would find how the supplementing of diet with royal jelly can change a normal female into a sexually viable egg-laying female that we call as the queen bee. Let us spend some time on the type of parthenogenesis. Viewers, normally a parthenogenesis should produce exclusively females because it was a female, but then here we find that there is a different type of parthenogenesis, and this is called as erinotoky. So, erinotoky or erinotocus parthenogenesis is also a type of parthenogenesis, but here the unfertilized egg is developing into a male and not a female. So, in other words, when it is developing into a male, this type of erinotocus parthenogenesis is a means of reproduction, all right, but then it is also a means of sex determination. And on your screens you will find a summary chart of how the queen behaves and how does a worker who is also deployed and a female also works to give rise to the erinotocus male. There is another very important aspect, and that is of the metabolically controlled sex determination. Now, in this case we find that we are not talking about XY or XX or ZZ or ZW. We are also not talking about the haploid or diploid type of sex determination. What we are here interested in to find out is that what is the relationship between the metabolic status of an organism and its sex. So, a scientist by the name Crew was the first person to have suggested that the metabolism also is a very important aspect which will tell us as to what is the balance between the anabolic and the catabolic status which would give rise to a particular sex. And Shull and Whitney have also demonstrated that if there is an increasing metabolic rate, it would increase the maleness in case of rotifers. So that means the kind of hormones, the kind of metabolism is also important in the determination of sex. Scientists by the name of Riddle found out that metabolism will play an important role. As I said before, if it is increased, then it would result in maleness, and if it is a decrease, it would result in femaleness of a particular organism. Let us take some examples as far as the metabolically controlled sex determination is concerned, because you will agree, learners, that hormones are going to play a very important role in sex determination, and this is actually seen in rotifers, arthropods, annelids and achuroids. We are going to take an example of Bonillia viridis, which is incidentally a achuroid. And Bonillia viridis is a marine organism. It's an achuroid worm, and you will find that there is such a large disparity between the size and the internal structure of an adult female and a male. The adult female is a fairly large one, ranges from 2.5 to 10 centimeters, and it has a very complex internal structure or anatomical organization. In sharp contrast, the male is very small, in fact microscopic, of the size of a protozoan, and it has very rudimentary organs. On your screens, viewers, you will find that the male, we've contrasted the sizes of the male being 3 mm and the female being 10 centimeters, and the proboscis of the female could extend to 1 meter, and the male Bonella, it lives symbiotically inside the reproductive organs of the female as a parasite. So that means the male is going to live within the uterus of the female, and you can see on your screens the enormous disparity as far as the size is concerned. Actually what happens is, after fertilization of the egg, the larvae are released in water. The proboscis is going to secrete a hormone like substance, and this substance is going to suppress femaleness, and once the femaleness is suppressed, then the maleness is initiated. So it depends. All the larvae are genetically and cytologically similar. If the larva falls down in isolation, it becomes a female in water. But if by chance the larva settles on the proboscis of the adult female, then the hormone would suppress proboscis secretions will suppress the femaleness, and it would result in the formation of a male. So no chromosomes are involved in this case. It is only the proximity or the distance which is going to result in the sex determination. And sometimes if incompletely developed male by chance detaches from the proboscis halfway through, then it would never become a male or a female, it would remain intersex. So this is how the sex determination works in Bonella. A very interesting example is also found in croaking hen, and this was discovered by the scientist Crew. So it is also called as a case of Crew's hen. Now interestingly, you would find, viewers, that there can be a complete sex reversal in case of fowls, because only one gonad in the female develops into ovary. The other gonad is there, but then just one of them develops into an ovary. If somehow the functional ovary is damaged, or a time comes when the natural egg laying stops, then what would happen? The rudimentary gonad which was left is not going to develop into the second ovary. It would now grow and develop into a testes. So that means the female sex can be reversed into a male sex, and fertile chicks now will show 2:1 ratio of females and males, and we know that the female in case of fowls is heterogametic. So during the development, what happens is the pituitary is stimulated to produce female hormones by the ovary, and by this time the male characters are suppressed. If somehow the ovary is removed, then the adrenals would become active, and then this would help in the development of testes. Another very classical example, which is also, I think, one of the most important from the examination point of view also, is the function of hormones and the origin of free martens. Free marten is actually nothing but a sterile female mammal with masculinized features and non-functional ovaries. So we find it is very, you must have seen even on Indian roads and other many of the cows moving around who look like females externally, but are perhaps not females to that extent, that is, they are not sexually viable. So in cattle it is very common, and actually they are intersex. So it is found that if there are fraternal or dizygotic twins which are formed, and the two sexes are occurring in the same mother, the female member is usually a sterile intersex, and this is called as a free marten. The case of free martens were first seen by the ancient Rome's greatest scholar Marcus Terenius Varro, and it was as early as in 27 BC. He gave the term taura. And the 18th century Scottish physician John Hunter found that all the free martens always have a male and a female twin. So what would happen is that these intersexes or the free martens, externally and genetically, they are females, that is XX. But then the external female genitalia are present, but there are no internal sex organs. that means the uterus is not there, the ovaries are not there, and the internal sex organs are like a male. What has really happened is that they have been sterilized in the womb itself, in the situation that the hormones for this function came from their male twin, and the male twin is normal. So what really happens is that the fetal membranes of the twin calves, they fuse, and this permits cross blood circulation among them. The male hormone, since it is synthesized earlier, it goes to the female womb and it suppresses the differentiation of female internal sex organs. This type of a situation is also seen in sheep, in goats, and also in pigs. It is very common, the free martins. On your screens, if you can see the diagram of the two twins, the male is normal. The female is also showing teats in the sense that it is showing external genitalia, but then and other structures, but then it is a sterile or a free martin. In viewers, in our next lecture, we will now spend some time on a distinction between genotypic factors which are responsible for sex determination and the environmental factors. As you can see on screen, till now we have discussed the left hand part where the genetic factors seem to be outweighing the environmental one. But then what happens if there is an influence of temperature, salinity, photoperiod, pH, or even the density? Even these environmental factors can result in sex reversal of the established XYZZ or XXZW, that is XYXX and ZZZW type of sex determination. So for this discussion, we have also used the references of Pierce, Klug and Cummings, and various other standard books like Strasburger, Strickberger, P.K. Gupta, and Ainsworth, along with the other genetical treatises. Thank you.